स्लाइड चेंज हो गई है यहाँ से।
मैडम टाइटल स्लाइड पे रखो।
हाँ जी, नहीं वो चेक कर रहे थे सर की स्लाइड
Good
चेंज
morning to all. Welcome you all in ISFCP dialogue series under the aegis of IQAC-IIC ISFCP, organized by ISF College of Pharmacy and Autonomous College Moga, Punjab. Today's guest and speaker, Ms. Richa Puri, and the title of her talk is ICH Guideline with emphasis on Q1 and Q2 guidelines. We welcome you, Richa ma'am. Please proceed.
Thank you, sir. Thank you, Mahan, sir. Good afternoon, everyone. And I'm going to present today ICH guidelines with emphasis on Q1 and Q2 guidelines. Let's talk about ICH guidelines. ICH guidelines, we also say that International Council for Harmonization or International Conference on Harmonization, it both goes side by side. What it provides, what is its role in pharma sector? International Council on Harmonization provides technical requirements for pharma sector. What kind of pharma sector technical requirements we are getting from ICH guidelines? They provide guidelines on impurity testing, GMP, stability studies, relevant threshold impurity testing, and many others like pharmacopoeias. They provide information on every topic, whatever is required in pharma sector or pharmaceutical industry, as well as pharmaceutical manufacturing, packing, dispatch, every sector. There is International Council of Harmonization is required. Let's talk about Q series guidelines. Q series have guidelines on Q1 to QF, but we are discussing about Q1 and Q2 today. Q1 is stability studies. What are stability studies? Stability studies provide the evidence on how the quality of the drug substance or product varies with time. Varies with time, but about how it varies. It affected by temperature, humidity, light, climatic zone. All these factors affect the product life as well as deteriorate the product with time. So for that calculation, for that judgment, for retest period, for shelf life calculation, we do stability studies. Stability studies provide the data for the product which can be tested throughout its shelf life period, or it is post manufacturing studies or post commercialization studies. But we do stability studies before registration of a product, or we collect the data so that we can register the product to make it commercial or make it in use for patients or public healthcare or in public sector welfare. What stability studies are? It consists of stability testing, or means like we can study the product throughout its shelf life with temperature, humidity, light, and climatic zones. Temperature, humidity, light, these are normal aspects. What are climatic zones? Whole of the world is divided into 5 climatic zones: zone 1, zone 2, zone 3, zone 4a, zone 4b. For that, if we are manufacturing products for export purpose or for domestic purpose, we have to check the climatic zone of that country, particular country for where we are selling our product. For that case, we have to check the climatic zone, in which zone we are sending the product. We have to do the stability studies for that product, particularly for that particular zone where we are selling the product or exporting the product, or if we are selling it domestically, we have to prepare it according to the climatic zone of India. India lies in zone 4b and zone 3. What are long-term stability studies? Long-term stability studies are those studies which are studied up to its shelf life, up to the shelf life of a product, whether it is 24 months, 36 months, 18 months, or 48 months, or whatever the requirement to maintain that particular shelf life. And accelerated studies are studies which are done for 6 months only. For 6 months, in this we do accelerated temperature, accelerated or we can say elevated temperature or elevated humidity conditions. We have a chart over there. These kind of temperature requirements are there for zone 1, zone 2, zone 3, 4, 4a, 4b. Also we do refrigerated conditions where humidity is not there, but only temperature conditions are maintained. Refrigerated conditions are also required for those products which are temperature sensitive, like nicorindil. We can do that. We can preserve that sample in refrigerated condition or in frozen condition to check that the stability study of that particular product. Now, guideline is Q1A-R2, that is stability testing of new drug substances and product. We can check the stability studies for new drug substances and products for registration purposes or filing the file in ICH guidelines or for commercializing the product after clinical activities have done. Next is Q1B, photostability testing. Photostability testing, as the name suggests, photostability means light sensitive products. For light sensitive drug products, light sensitive drug substances, or the drug products which are exposed through their primary packing. Primary packing, for example, we can say blister packing. The upper layer of the blister is PVC, which shows the product, make the product visible. In that case, if the product is come in contact with the light, it may get deteriorate. So for that case, we do photostability studies, which gives us idea, rough knowledge, or fair knowledge regarding what kind of the storage conditions we require for that particular product. That primary pack can also affect, marketed pack can also affected by the light, so that we can store the product in proper manner, and it gives the precautionary measures while manufacturing of that product. While manufacturing, it does not encounter with the light, so that the product may remain stable during its manufacturing and throughout its shelf life. For that, we need to check the what kind of label or what kind of primary packaging we have to provide for that particular case. We can use LULU or strip as well for that light sensitive products. Not We can avoid blister packing as well. Next, our third guideline is Q1C, stability testing for new dosage forms. New dosage forms are there. Different means new dosage forms are there, like we can say different pharmaceutical product with different type of active substances. Sometimes same type of active substances, which include different route of administration. We can use oral, parenteral in both case, with same active substance and different dosage form, or different active substances with different dosage forms. Sometimes we can include new specific drug delivery systems like modified release, extended release, immediate release, delayed release, sustained release, these kind of new dosage forms. And also we can include in this stability testing for new dosage forms is different storages, different storage forms with same administration route. Same administration route we can conclude in it, like tablet, like capsules, like suspensions. All the three are for same oral route, but dosage form is different. For this, Q1C provide guidelines for same active substance with different dosage forms or different oral different routes. We can say oral route or parenteral route, emulsions. They come under emulsions, suspensions, syrups, tablet, capsules. They come under oral route. For parenteral, it might be intravenous, intramuscular, subcutaneous. Whatever the dosage will be there, but dosage forms are different. Next is bracketing. Q1D is bracketing and matrixing. Sorry, bracketing. What it shows, bracketing, like we know the extremes. Extremes. Some like I have given the example of a torvastatin. If we have manufactured in the same manufacturing plant at the same site, we have manufacturing a torvastatin with different strength. Which what it does? It causes higher number of batches, which can go under stability studies. For strength, atorvastatin 10 mg, if we do manufacturing of atorvastatin 10 mg, there are first three batches which have to be charged in stability. After that, if any other validation batch or annual batch comes, we again have to go for stability studies. What it will do, it will increase the stability samples, includes the increased, manpower increased, and time So to overcome these factors, if we are preparing atorvastatin 10 mg, atorvastatin 20 mg, means the active product is same, manufacturing method is same, and formulation is same, like dosage form is same. We are preparing just atorvastatin 10 mg tablet is there for every strength, like atorvastatin 10 mg, 20 mg, 40 mg, 80 mg, 120 mg, as I have mentioned in the slide. If we are preparing same drug product or same API in same dosage form with different strength, having different having same manufacturing formula or method for formulation or same packaging material, everything is same, then what we will do, we go for bracketing. What will cause, in this we goes for extreme lower strength or extreme higher strength. For that case, in bracketing we consider extreme lower and extreme higher case. Extreme higher strength, atorvastatin 10 mg and atorvastatin 120 mg is selected, and only these two strengths will be charged for stability studies. Only these two will be charged for stability studies. What is the principle, or what is the main reason behind bracketing, is to save the manpower, the resources, and the time. After this, we can conclude the stability data for all the intermediate strengths. It gives a judgment regarding both the products, both rest of the strengths, but it provides data only for extreme sites, not for the intermediate sites, which we can judge, or we can get the data as soon as possible and with less time effect and with less manpower and less resources. Next is matrixing. In matrixing, we select a subset of possible samples tested at specific interval. In this, we can apply it to different batches, different strengths, different size of same products, or same container closure system. What we will do, as we have to perform stability studies at different time intervals, and those time intervals are third month, 6 month, 9 month, 12 month, 18 month, 24 month, or upwards shelf life, whatever it is. Stability studies will generally be done up to shelf life and plus 1 year. 1 year is for checking the product degradation after its shelf life. Shelf life is also generated or detected or presented with the help of stability studies. Stability studies provide the complete data of 24 month, 36 month, 48 month, whatever we are preparing the list. They will provide us the data for stability studies of that product on that particular data provided by stability will will maintain or will help in preparing its shelf life. Whether we have to give 24-month shelf life, whether we have to give 36-month shelf life, it will be provided by stability data, or it will be maintained by stability team. In this matrixing, what we will do, for some products we do matrixing. In this, selected subsets of possible samples are tested at specified intervals. For this, as I have mentioned over here in the slide, like third month, 6 month, 9 months, the test parameters are there. First is time intervals and another is test parameters. Test parameters which are generally performed in QC are dissolution, disintegration, uniformity of content, assay related substances. All these tests are qualitative and quantitative tests, which give the data regarding the product, what we have charged or what we have prepared. They are providing the stability data. For this, we will select some subsets. Subsets means some selected tests which we have to perform at different time intervals. Like I have make a tick mark over here in the slide, as at 3 months we are just doing dissolution profile, assay related substance only, and at 6 months we are doing dissolution profile, disintegration time, uniformity of content. So like this, we have to select some of the tests at some time intervals, so that we don't need to perform every test at every single time, every single interval. It will reduces time, manpower, and resources as well as. Next is evaluation of stability data. What about it? It is Q1E. It proposes the retest period and shelf life evaluation. Shelf life evaluation, like determining the shelf life or expiry date of the product, or retest period, when we need to retest the same product again, or at what time we will do the next quality control analysis. Also, it addresses the drug that should be submitted in registration application for new drug substances. It provided the guidelines which we have to do at the time of the evaluation of stability data. Also, it give guidelines for generating the stability data which have to be submitted while filing or registration or submitting the data. Q1F was also there, but it was withdrawn by ICH guidelines. So they will update that, and later on we will discuss about that also. Next is analytical validation, that is Q2. Analytical validation. What is validation? First of all, validation is to produce a documented evidence. Documented evidence, or a written evidence, or what we are doing validation is for specific process like manufacturing, compression, granulation, sieving, milling, coating, punching. For that, these are specific processes. And equipment, for every equipment there goes validation. Validation of equipment is called as qualification validation or equipment validation. In this, there are processes like concurrent validation, retrospective validation. This goes for equipment validation. Method or system consistently produces a result meeting predetermined acceptance criteria. Whatever the acceptance criteria has been set has to be accomplished by the validation protocol. That validation protocol gives you the subsets of the test we have to perform during that validation procedure, like specific processes and equipments, method, system. Everything has to be validated. Whatever the protocol or procedure mentioned, we have to follow that to meet the acceptance criteria. If it's meet the acceptance criteria within the limit, it is validated. Now, what is analytical validation? As the word suggests, analytical. Analytical goes for analysis, and analysis is done only in quality control lab. What are the what is validation? Analytical validation. It is the process used to prove the analytical procedures employed are fit for their intended use. Means it gives the information that the specific instrument is available or is uh, ready to use, or it will give the intended results whenever it is used. If any of the instrument is not validated, it is mentioned as under validation, under calibration. These kind of labels are already presented over the same equipment which is not yet validated. It consists of qualitative and quantitative tests. What are these tests? First is specificity. Specificity, in this specificity, what we will do, we check that the same product we are doing the analytical validation with the same sample, or same whatever the product we are using, like glimepiride, metformin, which of the drug product or API we are using. In this, it shows in this we perform specificity. Specificity goes for identification of the same product, as if we are doing the analytical validation for metformin, we are doing the analytical validation for glimepiride. It gives identification regarding that same raw material or API. We will do purity test, and we do assay. Assay is checking whether the claimed drug is present in that sample or not. Next is linearity. In this linearity, the concentration of the sample should have to be directly proportional to the absorbance it is provided. It is providing. What kind of absorbance it is providing? As we are doing UV analysis or HPLC analysis, it shows the results accordingly. Like I have plotted a graph here. X-axis is concentration and y-axis is absorbance. It shows a linear graph with standard deviation of 0.99, which is equivalent to 1. and the equation is there, y is equal to mx plus c is constant here. And linearity, in linearity we have to prepare the 5 concentrations like 25%, 50%, 75%, 100%, and 125%. We have to prepare different concentration and check different concentrations at same time, so that it provides us the data whether that a product usually claims to get 100% of the claim. If we are checking the same product at different concentration, does it give the same result or same accurate result as we require, or as we are getting it 100%? For that, we have to split the concentration in 5 different manners, that is 25%, 50%, 75%, 100%, and 125%. At different concentrations, we will check the absorbance, whether it is giving accordingly or not. This will be checked, and in concentration and absorption, there will be a graph which has to be plotted like this, what I have plotted over here. Third step is accuracy. Accuracy generally suggests that the closeness of the results to its true value. In this accuracy, what we will do, we will take a mixture of excipients, or like we have a complete drug formulation, but of a tablet. We discuss about any kind of tablet. We discuss about telmisartan tablet. Here we have, in that telmisartan tablet, we have API and excipients, blend of both API and excipients, but that is complete tablet. In accuracy, what we will do, we will take just single blend of all the excipients at the same concentration in that particular tablet, whether it is 10 mg tablet, 40 mg tablet, 80 mg tablet, whatever the strength there would be, we will take the blend of excipients. At same time, we will spike the concentration of the API present in that particular tablet. We will spike that drug over the excipient, and then we will perform the analysis. Like we do assay, it is similar to somewhat similar to assay procedure, what we will do for determining of that same product in that particular tablet. Here we will produce 3 to 5 concentration, like 75%, 100%, 125%. In 75%, what we will do, we will pour the drug, or we will spike the API into the blend of excipients, 75% of the claim of that tablet. In 100%, we will spike 100% equivalent to that tablet's claim. Whatever the claim there would be, we will spike the API into blend of excipients what are present over there, equivalent to 100%. And in third concentration, we will pour, or we will spike 125 mg or 125% of the drug into blend of API, blend of excipients. Likewise, we will prepare 3 concentrations, and after this, we will do it in triplicate. All the 3 concentrations will be performed in triplicate, and the result will be calculated on over their basis. Next, our parameter is precision. In precision, what is there? There are 3 kind of tests which are performed. First is repeatability, second is intermediate precision, third is reproducibility. In repeatability, what we will do, it should be performed within the same lab, but with different analysts at different time. 2 analysts will perform within same lab at different times. What is intermediate precision? It has to be performed the same test, same analytical parameter within a lab at different days with different analysts. 2 analysts will perform, but at different days. What is reproducibility? It is reproducing the same results which has to be performed in different labs. 2 labs will be considered, and in those 2 labs we will produce the same data with different analysts at different time. This is reproducibility. Fifth is limit of detection. Limit of detection is lowest amount of the analyte or API or we can say lowest amount of analyte which is present in the sample and can be detected but not quantitative. Means we can detect the presence of that sample, that particular analyte in the sample, but we cannot quantify that. It can be calculated with standard with signal to noise ratio and standard deviation of the response and slope. We can calculate limit of detection, or it is also called as LOD. LOD value has to be calculated by using standard deviation of the response and slope. Next is signal to noise ratio, which can be calculated with the formula 3.3 multiplied with standard deviation divided by slope. This all can be calculated easily. Next is limit of quantification. Limit of quantification is the amount of analyte in the sample which can be quantitatively determined, or it has the same formula to calculate it by signal to noise ratio, that is 10 multiplied by standard deviation and divided by the slope. This is limit of quantification. Next is robustness. It is the method to check or measure to check the capacity of a sample to remain unaffected with small but deliberated variations. Whatever the variation we will do, that should not cause any kind of drastic variation in the sample. Parameters which can be affected, like pH of mobile phase. We can, as we have suggested to prepare the mobile phase with 7 pH, we can change the pH with 6.9 and 7.1. These two changes may cause small changes but should not be deliberately variable. Likewise, composition, we can make differences in the composition while preparing mobile phase or any other sample. Mobile phase variation should be there, temperature variation should be there. Wavelength change, as we have to suggest that we have to perform metformin at 284 nanometers, we can change the nanometers or wavelength by plus 1 or minus 1, like 283 and 285 nanometer. We can check the same sample at different absorbance whether it is giving the proper results or not. Solution stability, it can be checked at 0 hours, 6 hours, 18 hours, 24 hours, so that we can check that the same sample has to which is we are using for analysis is stable for 1 day or not. For that basis we can calculate whether it has to be preserved or not for 1 day, or it has to be freshly prepared or have to discard it by the same day. Last one is range. It gives the interval between the upper and lower concentration of the analyte and the sample in different analytical parameters. This is all about Q1 and Q2 guidelines. Thank you.
Thank you,
Okay,
Richa
sir.
Puri ma'am, for this wonderful information.
Okay, sir.
And we are very thankful to all listeners for your precious time. If you have any query, please inbox your query in the chat box of this YouTube channel, and please also visit our website www.isfcp.org. Again, we thanks to all, and thank you, Richa. Thank you so much.
Thank you.